we've talked about light reaction. Okay, so in photosynthesis, light reaction is just basically the first 50%. Okay, it's basically just the first 50%. So we still have the next 50%. So the next 50% is basically your, let me open your slides, is your dark reaction, or sometimes also known as your Calvin cycle. Okay. Okay, so also known as carbon fixation. So you can find all the materials inside the ellipse. So carbon cycle. Sorry, okay. So from the first half, we talked about light reaction. So from the light reaction, you already know, you should know, okay, from where is your oxygen produced, okay, and who is the final receiver of your electron, okay, and what is produced is nonetheless your ATP and also your NADPH. Okay, so these two are the important players that comes to play in Calvin cycle. Okay, so these two are more like the requirement, okay, the things needed. But actually, for your information, the real pegawai or the officer that is in charge of controlling this Calvin cycle is very much the enzymes. The first enzymes that catalyze the binding of the first, because here you see ATP and NADPH are basically the things that are needed, that is needed as kind of like benda yang diperlukan lah semasa proses Calvin cycle. But in order for this to kickstart, this carbon dioxide needs to come in and bind with the number one player inside here, which is your ribulose 1,5 biphosphate. Okay, so the binding of these two, okay, is actually kind of like under the care or under the attention of this enzyme called Rubisco. Okay, we will come to that in a moment, and you will know Rubisco is actually the main player that is controlling this whole process. Jalan ke tak jalan? Okay. So the reactions are powered by ATP and NADPH. Okay, so the fixation of carbon dioxide and the synthesis of carbohydrates occur in the cytoplasm of bacteria and in chloroplast if we are talking about plant. Okay, so it is inside cytoplasm if we are talking about in bacteria, but it's basically in stroma, which is why light reaction happens at the thylakoid. Okay, your granum. Okay, then this is your stroma, the outer environment. So your ATP are generated outside here, and your NADPH. So they are used inside the stroma because your Calvin cycle takes place inside stroma. Okay. Okay, so there are several names like what I mentioned. Some call it Calvin cycle, some call it C3 pathway, some call it reductive pentose phosphate cycle. Okay, or some even call it dark reaction. Okay. So next, okay, the Calvin cycle, basically this is the cycle itself, can be divided into three main components. Firstly, oscillation. Okay, some reference also call this fixation because this is the first step. Okay, your step one, where ribulose 1,5 biphosphate is being bind together with carbon dioxide. This ribulose 1,5 biphosphate is a 5-carbon compound. Okay, 5 carbon, contains 5 carbon. Once it binds with your carbon dioxide, it is going to form a 6 carbon, 5 plus 1, 6. Okay, so some call it carboxylation, some also call it fixation. okay, the fixing of carbon dioxide into your ribulose 1,5,5 phosphate. This fixing process is catalyzed by an enzyme called ribulose. Okay, so in a moment we will look at that enzyme lah. Okay, so after fixation, it is gonna be 5 plus 1. You end up becoming 6. Very fast, this 6 is immediately gonna be chopped into 2. Okay, you end up getting 2 molecules of 3-phosphoglycerate. Okay, so I've got pictures. Okay, this one. Okay, okay, so you see, this is your ribulose 1,5,5 phosphate. Okay, under fixation process or carboxylation process, your carbon dioxide comes in. Okay, these are some of the intermediate that is gonna form in between. Okay, so with the help of Rubisco, the carbon dioxide is gonna come in. Okay, it's gonna bind, form this, and with a series of process, okay, involving water some more, you are going to end up getting 2 molecules. So this 6 carbon is gonna chop off immediately, very fast. Okay, forming 2 molecules of 3 carbon. 1, 2, 3, 1, 2, 3. 1 molecule, 2 molecule. Okay, both of this is going to be the same. So 2 molecule of 3-phosphoglycerate. Okay, so this is the carboxylation process. Okay, so you end up getting 3-phosphoglycerate. Two of this. Because you have 6, you chop it into half, you have 2 molecules of 3 carbon. Okay, then with the help of ATP, now this is the part where the ATP that you generate from the light reaction is going to be used. Okay, so it is going to be phosphorylated. That's why you see from 3 phosphor, now you end up getting another additional. So the numbering normally tells you the location where the phosphorylation takes place. Okay, so one of the phosphate group here is going to be cabut and ditampalkan. Okay, it's being glued onto the first position. That's why you have 1,3 by phosphoglycerate. Then your NADPH is going to come in again. Okay, so now you already use ATP. Now you already use NADPH. So these two are the one produced from your light reaction. So it's going to be used inside your dark reaction. So this one under a process called reduction. Okay, so this one, your NADPH is going to reduce your 1,3-phosphoglycerate into glycerol 3-phosphate. So this is basically the final component inside your carbon cycle before this is converted further into sugar. Okay. Now, as you can see here, this is supposed to be a cycle, okay, which is why they call it Calvin cycle. So it needs to go around. So generating this, if you were to finish all of this for glucose formation, then who is going to produce ribulose 1,5-biphosphate again to bind for the carbon? So in order to generate this ribulose 1,5-biphosphate again, okay, so that more carbon can be fixed, okay, to produce glucose, some of your glyceroldehyde 3-phosphate, G3P, okay, is gonna be channeled. Okay, it's gonna be channeled for regeneration purpose. Okay, so normally in this whole cycle, you have 3 main steps. First, you fix, the carbon comes in, then you reduce it further by using up the ATP and NADPH that you got from the light reaction. And after that, don't forget to regenerate back this compound. Okay, without this cannot be fixed. Okay, so you have to regenerate. So this basically, okay, this is the person, okay, who kind of like showed the process, which is why the name, they kind of like remember this contribution by naming this, or maybe it's kind of like this, the normally researchers, they tend to name things after themselves, or it could be people want to appreciate his or her contribution. So the process is named on the discoverer lah. Okay, so now this sentence here, okay, so like what I mentioned, Rubisco is the main player. Okay, it's the most important one. So you have this and this. These two needs to come together, but whether it comes together ke tak depends very much on an enzyme, okay, in between. It's not being shown in this picture, but you have this enzyme called Rubisco. Okay, Rubisco catalyzes the fixation of atmospheric carbon dioxide into carbon compound. So basically, you imagine Rubisco to be a person grabbing carbon dioxide from the atmosphere, then glue it with your ribulose, uh, ribulose 1,5,5 phosphate. Okay, so another name for your Rubisco is actually ribulose 1,5,5 phosphate carboxylase oxygenase. Okay, carboxylase oxygenase. Okay, you will come to know in the next few slides, this Rubisco not only can glue carbon dioxide to your ribulose 1,5,5 phosphate, okay, it not only can glue carbon dioxide, it can also glue oxygen to ribulose 1,5,5 phosphate, which is not preferred. Okay, tak nak. Kalau boleh, jangan. Something like that. Okay, preferably Rubisco is intended to bind carbon dioxide so that we have carbon fixation, so that carbon cycle can jalan, boleh jalan. It's not like you bind oxygen then tak boleh jalan. There are certain weakness for that. Okay, so which is why the name here for Rubisco, you have ribulose 1,5,5 phosphate, which is basically the substrate that catalyzes. You have carboxylase and oxygenase. Adding of carbon dioxide is carboxylase, while adding oxygen is oxygenase, meaning to say this enzyme can do both. Okay, but the preferred one, okay, is of course carboxylase. So it depends on the condition. Okay, we will explain more of it in a moment. Okay. So what actually happens is the carbon dioxide is combined with the 5-carbon sugar, okay, which is 1 to form a 6-carbon intermediate, okay, which leads to formation of 3 carbon molecules of 3-phosphoglycerate, 3PG, okay. Whenever you go through the materials, if you come across a video link like this, do click on and have a look, because this video normally shows nice animation, which is good for you to have an idea what actually happens, because to me personally, I like the animation, so it makes understanding better. Okay. Okay, so now this is the structure of your Rubisco. So it's basically very big, containing 8 large subunits and another 8 small units. So you can imagine it's basically a huge complex consisting of 16 subunits. Okay, 8 large, 8 small. So the main important component, the active site, enzyme, when we talk about enzyme, there's definitely active site. So for Rubisco, the active site is at the large subunit. Okay, so active site is associated with the large subunit, and this large subunit is encoded by the genome in the chloroplast, whereas the small subunit is encoded by the genome inside the nucleus. Okay, so in plant, okay, you can actually find up to 50% of this Rubisco. So you can imagine how important. You see, when you pluck a leaf, right, from a plant, then you extract all the protein out, you will actually find up to 50% of the total protein inside the plant leaf is actually Rubisco. So you can imagine how important is this enzyme, how much the plant needs this enzyme for photosynthesis. Okay, so far from research, we have only discovered the property of this Rubisco to be involved in photosynthesis. Who knows, there are actually many more function that this enzymes might be involved in. So far, uh, a huge portion of this was found, and it indicates its importance. Okay, if it's not important, for sure the plant will not spend so much energy in synthesizing this Rubisco. As you can see, it's so huge. Yeah, you can imagine how many genes are involved to actually synthesize and have that huge protein coming out. Okay, so in order for this Rubisco, as you are aware, Rubisco is the player that kickstart, right? Okay, so kalau ribulose 155 phosphate, tadi, kalau if this wants to bind with this, then it depends on it needs the help from Rubisco. Okay, so here in Rubisco, this is what happens or what needs to happen in order for the reaction to take place. Okay, so in Rubisco, now basically you imagine this to be the active site. Okay, when you talk about enzyme, you have to imagine the active site. So normally, you have a carbon, right? No, not carbon, sorry. You have an enzyme. Okay, imagine this is an enzyme. So enzyme, normally you have the active site. Active site is basically like a lubang, like a hole. Okay, a hole for the enzyme to grab hold of the substrate. Okay, so in the active site, this needs to happen. Okay, so your carbon dioxide needs to be glued onto your ribulose 155 phosphate. Need to be glued here. Your carbon dioxide kena masuk dalam sini. Okay, so but before this, this is being put into this, Rubisco yang akan bind this first. Okay, so how? By forming this structure, which is called this adduct, carbamate adduct. Okay, this carbamate adduct. Okay, so this is the side chain. So you have your Rubisco, you have amino acid, right? Proteins are made of amino acid. One of the amino acid, lysine, okay, is gonna have some protruding side chain. Okay, so this side chain here, especially this amino group, is actually the part that is gonna bind to your carbon dioxide. Once it binds to your carbon dioxide, your Mg2 plus comes in. Okay, now this is the part, if you recall from our first part of our lecture, Mg2 plus needs to be concentrated inside the stroma. Okay, which is why this Mg2 plus is needed. Okay, it is needed, so it needs to be accumulated inside stroma. Okay, you still remember the one that I mentioned? Stroma is being Mg2 plus is being pumped out of the lumen while chloride is going in. Okay, so your Mg2 plus is needed for the first reaction. Okay, so you form this carbamate structure. Okay, carbamate adduct. Okay, so once you form this, okay, now this, okay, sekarang ni this Rubisco dah pegang carbon dioxide, so it can then grab on to your ribulose 155 phosphate to glue it together. Okay, and is to form the bond lah. So I tend to use the word glue so that you imagine it's sticking together lah. It's like putting pieces of Lego together. Okay. Okay. So in the dark, carbamate adduct dissociates. Okay, from the active site. So which is why okay, that reaction, some references what might when you find on Google, right, you might come across sentences whereby it tells you uh, light reaction you need light, okay, which is correct. Dark reaction you don't need light. Okay, that is, I would say, partially because without light, okay, as you can see in the dark, carbamate adduct dissociates. Okay, without light, this 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 structure here actually dissociates. So it tends to kind of like break apart without light. So without this, how can your Rubisco bind this carbon dioxide to ribulose 155 phosphate? Okay, so this actually in the dark, this process actually is very much difficult to be carried out. Okay, because without light, this structure is difficult to be formed. Okay, so now this is the things that we saw just now. Okay, let me just finish this part first before we go in further with this. Shall we stay? Should I go for this one first? Yes, another information. Okay, so in continuation from that slide just now, okay, so from here, so because we talked on light, right? So in the dark, this thing do not actually form. Okay, it's kind of like dissociates lah. They form, tapi it tends to break up easily. And then we have this fabulous 1,5 bisphosphates that comes in and block the whole thing, which then inhibits the enzyme. So if Rubisco is inhibited, then you don't have any Calvin cycle anymore. Okay, so Rubisco activity is regulated by pH and G2+, okay, which is the one that I told you. Okay, in the light reaction, okay, so in the presence of light, if you recall in a previous lecture, this is your thallicoid. Okay, you have H plus being accumulated. Okay, you have H plus being accumulated while your Mg2 plus is being pumped out of your thallicoid. So eventually you have Mg2 plus. Okay, why you need this? Because Rubisco needs Mg2 plus. Okay, so this is being accumulated and you end up having, in the presence of light, you have the whole photosynthesis process to be able to carry it out. You have NADPH to produce, and you have your ferredoxin under a reduced state or reduced condition. Okay, this ferredoxin in a reduced condition is very much needed. Okay, it's very much needed because this actually is an enzyme. If you recall ferredoxin in the previous lecture, ferredoxin is the member just before passing the electron to your NADPH. Okay, NADP to form your NADPH. So this needs to be in a reduced form. So okay, now this okay now this one now. So in the presence of light, you know your from the light reaction, your ferredoxin is gonna be carrying your electron, right? So eventually it is gonna be in a reduced state. Once it carry electron, it is in a reduced state. So in a reduced state, it is then gonna reduce your NADP to NADPH. Okay, so we end up getting NADPH. But not only does your ferredoxin in a reduced form reduces NADP, it also reduces a protein called thioredoxin. Okay, it also reduces thioredoxin. So thioredoxin is an important enzyme that is needed to activate all the other enzymes that is needed in Calvin cycle, including your Rubisco. Okay, so which is why dark reaction don't really function well in the dark. Okay, so if it's in the dark, you don't have light, you don't have rhodopsin in the form which is reduced, so you don't have NADPH produced. And at the same time, you don't have rhodopsin to activate this protein called thioredoxin. This protein, thioredoxin, this one, needs to be activated so that it can activate Rubisco, okay, and some other enzyme. Okay, so which is why now these are some of the lists of enzymes that needs or depends on the activation of thioredoxin. Okay, includes Rubisco. Okay, so if thioredoxin is not activated, then Rubisco is not activated. If Rubisco is not activated, your Calvin cycle is not activated. Okay, so thioredoxin needs to be activated. So how does thioredoxin is activated? Thioredoxin is activated by reduction caused by ferredoxin. Okay, ferredoxin is activated from the light reaction. Okay, if you recall from light reaction, after photosystem 1, the electron is going to be passed to ferredoxin. Ferredoxin, once it receives the electron, it becomes a reduced state. Okay, it becomes pyridoxine reduced, RED. So this reduced pyridoxine is gonna activate thioredoxin. So besides activating thioredoxin, it also reduces your NADP, okay, which is dia ada dua kerja lah, okay. So which is why it kickstarts the whole thing. Okay, so reduced thyroid can activate enzymes, but it's so this is the mechanism. Okay, this is more towards the mechanism where you see this SHSH. Okay, so activated, so we don't have that sulfide bond. Okay, so same likewise, it all depends on the formation of disulfide bond. Okay, so the important thing is pyridoxine needs to be in an activated form or reduced form to activate thioredoxin. Once thioredoxin is activated, it can then proceed to activating all these enzymes. Okay, so you see, Rubisco is one of it, okay, which is very much needed for carbon fixation in the Calvin cycle. So if this enzyme does not work, your Calvin cycle will not start. Okay, so that is more towards the regulation. Okay, how is it controlled? Okay, so now let us look at the Calvin cycle itself. Okay, now you are aware that here, okay, this needs to bind to this. Okay, binding is assisted with the help of Rubisco, okay, the enzyme. Okay, so the process you have already seen this one. This process is called carboxylation, catalyzed by Rubisco itself, capable of doing carboxylation. Okay, carboxylase. Okay, you end up getting uh, two of this molecule, 3-PG. Okay, 3-phosphoglycerate. Now this 3-PG is gonna move on to reduction process. Okay, reduction process, which is this one. Okay, conversion of your 3-PG to glucose is very similar to your gluconeogenesis. Okay, which is the process of glucose formation inside animal cells or eukaryotes. Okay, but the difference is this one. Look at this one. Okay, so glycerol dehydrogenase in plant you use NADPH. Okay, you need NADPH. So this is the part where your see the first part you consume ATP, and then the next stage you use NADPH. But in animal, in gluconeogenesis process, you don't use NADPH, you use NADH. Okay, NADH. But in plant, you use NADPH. Okay, this is the stage. Okay, so the steps require consumption of ATP and NADPH, which you have generated from your lab reaction. Okay, so so this 3-PG, okay, 3-PG. So sometimes if you refer to the name in textbook, for example, in figures, you will come across they just abbreviate this to be 3-P and G, 3-PG, 3-phosphoglycerate. Will also be exported to the cytosol and be used in normal gluconeogenesis. So it can be channeled out as well. Okay, it can be it can be channeled out as well. So exocysts can then be used for energy or starch synthesis. So eventually, as you goes on, this is the reduction process. You use up ATP, you use up NADPH, then eventually you end up generating G3P, glyceroldehyde 3-phosphate, G3P. Now this G3P is then capable to be channeled for various purposes. You will come across this G3P very often inside several pathways. Okay, so it's like the crossing, overlapping pathway, okay, of different process. Okay, so eventually you end up getting glyceroldehyde 3-phosphate. So it can be channeled for the formation of sugar. Okay, hexose 6-carbon sugar. Okay, so after you have generated this, okay, so normally from the process you have two molecules of this uh, this PG. 3PG, this 3PG, you have two of this because initially Rubisco, you have ribulose 1-phosphate, 1,5, you have ribulose 1,3, ribulose here. Okay, you have this, which is your 5 sugar, 5 carbon, plus 1, you become 6. Chopped, you will become 2 of this. Okay, 2 molecule of this is then going to be phosphorylated, reduced further, so you end up getting 2 of this. You also end up getting 2 of this. Okay, which is why in this figure you see all of this to be 2, 2, 2. Okay, so eventually this is going to be accumulated. Okay, then you end up getting your fructose, which is just 1. Okay, but bear in mind, not all the G3P produced is going to be channeled for the production of sugar. Okay, the next stage, which is your regeneration stage, you need to regenerate, okay, the the ribulose 1,5 biphosphate, okay, needs to generate this, okay, so that the pathway can be released, keep on cycling. Okay, one of the two 3-phosphoglycerates goes towards regeneration. Okay, it needs to generate 5 carbon sugar from 3 carbon, okay, and 6 carbon sugar. Okay, so basically the process is a combination between transketolase and aldolase. Okay, so it's very much expensive because you need a lot of ATP in the it consume some of the things needed. Okay, so now this is your regeneration stage. Okay, so now as you can see, part of it is being regenerated. Okay, while if you this one, you have two, right? One of it is being used, while the other is uh, one of it is used as sugar production, one of it is being regenerated. So how is it regenerated? So this is how it's going to be regenerated. You have this 6 carbon sugar with the help of a transketolase. So basically this here is going to be chopped. You chop here, and then this part, you stick it together with this. You end up getting this structure. Okay, so 5 carbon sugar. Yeah, 5 carbon sugar, and you also have the remaining of this, okay, which is a 4 carbon sugar. So this is a 6, 1, 2, 3, 4, 5, 6, 7, 8, 9. Okay, so this one is 1, 2, 3, 4, 1, 2, 3, 4, 5. Okay, 5 is 1. So 5 plus 4, 9. So in total here, 9 carbon. Okay, so this one here, okay, once it has been, this is basically the regeneration. How does the cell regenerate from this back to this ribulose 1,5 phosphate. So after chopping, you have these two components coming out, right? So this one here, okay, this 4 carbon is then going to be bind with this 3 carbon compound. With the help of aldolase, you end up getting this 7, 2, 3, 4, 5, 6, 7, yeah. 7 structure, which is then going to be removed 1-phosphate group. Okay, from the top here, removed, and you form this 7 sugar again. Then after that, you are going to have another. This structure is then going to be added with another G3P, okay, with the help of ketolase again to form your 5 carbon and another 4 carbon. 5, 5, 5 plus 5. Okay, so you basically end up generating 2 more of this. Okay, first one here, the second one here again. Okay, so these two are used. Okay, so it's not like they are not used. Okay, so this one here is going to move on to the next. Okay, so this one, okay, ribose 5-phosphate, this is one. Okay, whereas this one, cellulose 5-phosphate, this one is then going to be channeled, but with the help of different enzyme, it's going to be formed into ribulose 5-phosphate. And lastly, with the help of more ATP, is then going to be regenerated back to this one. Okay, because it's basically uh, a cycle. Okay, so as you can see, it is a cycle. So the regeneration part is more kind of like more steps lah. Okay, so as you can see, based on if we were to track based on the number of carbons lah, okay, eventually it's gonna rotate and generate back the 5 carbon here. Okay, so that when your carbon dioxide comes in again, you can form back that 6 carbon. Okay, so this is your overall formula lah. Okay, so this is the regulation. Okay, so just now we talked about light. Okay, so in the dark, so in the light, as you are aware, so we have the accumulation of hydrogen, Mg2 plus going out, you generate NADPH. This one, your pyridoxine is reduced. Okay, so that you can activate various kind of, uh, you can reduce NADP to NADPH, and at the same time, you can reduce your pyridoxine. Okay, but how about in the dark? So if in the dark, you don't have your H plus because you don't have photon to activate the electron transport. So you don't have electron transport, you don't have accumulation of H plus. So this one is not acidic. Okay, so your H plus is mostly outside. And your Mg2 plus is not being transferred. Pyridoxine, of course, without electron transport, you don't have this to be in the reduced form because no electron to be collected by pyridoxine. So at the same time, NADP is not being reduced. Okay, so all factors are influenced by light. Okay, so with light, no light. Okay, so this is your thyrodoxine, which is very much needed to activate the reaction. Okay, so now Rubisco, just now we talked about that this Rubisco here. mm, Okay, you see, the name Regulus 1,5-phosphate carboxylase and oxygenase. It is capable of binding carbon dioxide and also oxygen. Okay, so if you look at the process, okay, so why did I mention that the binding to oxygen is not preferred? Okay, because you are going to lose one of the uh, carbon. Okay, it's gonna cause the losing of one of the carbon. Okay, if you look at this one, under normal condition, the rate of carboxylation is carboxylation, so it's normally more. Okay, it's normally more the kind of like because the plant also knows that they are supposed to Rubisco is supposed to bind carbon dioxide, so they have this higher tendency to bind carbon dioxide still. However, they still can oxygen, but the rate is different. Okay, both require activation by carbamate adduct. So no matter whether uh, Rubisco binds to carbon dioxide or does it bind to oxygen, still requires the formation of that carbamate adduct that we talked about just now. Okay, it still needs to form this this one, this structure. Okay, so if this is not formed, okay, you see, if this is not formed, so the enzyme is basically stopped. Okay, so if it's stopped, okay, so if it is binding to oxygen, okay, so this is okay if it is uh I think nak tanya soalan kot. No, I'm just kidding. Okay, so okay okay, oxygen. Okay, so if so your Rubisco can also bind carbon dioxide and also oxygen, right? Can bind carbon dioxide and oxygen. So if binding carbon dioxide, then yes, everything goes well. Yeah, but if it binds oxygen, okay, so what happens is when it binds oxygen, this Rubisco is basically gonna add this oxygen and form this this process. Okay, so if you recall in this, so here's okay this one. Okay, basically this is the same as the picture just now. Okay, so if with carbon dioxide you form two of this 3 Pg, 1, 2. But with the presence of oxygen, you only form one 3 Pg. You have additional one structure that is called phosphoglycolate. Okay, so there is already a form of loss here. Okay, instead of forming two, in this Calvin cycle, one of it goes to, because these two actually goes down further to the reduction, right? Goes down to reduction to generate. You see, this two will go down to your reduction process to generate your this one, two of this. Okay, so two of this goes to two of this, and then eventually one goes to generate the sugar, another one goes to regenerate the Rubisco, right? So if you have oxygen that is binding, so if you have oxygen that binds to Rubisco instead, you only get one this, you only get one of this. Okay, so if you only get one of this, that means you also only get one of this. So more cycle needs to run in order to gain sufficient amount of your G3P so that you can form Rubisco again, and some more you have to divide to regeneration. So rugi lah. Okay? But in plant cell, normally that is the difference. So that is the difference between carboxylase and oxygenase. So even though that this is generated instead of two of this molecule, this in the biological world, nothing is being put to waste. Okay, so this here, okay, if we go to this one, okay, see, we generate this one, right? So what happens to this? So this is not being put to waste. This one goes into the normal process, okay? But this one here, okay, so you see, you generate this one. Okay, what happens if oxygen starts to bind? You generate this. Okay, one of it, your normal 3 Pg is gonna go continue your carbon cycle lah. But tapi rugi sikit lah. Instead of getting two, you only get one. So this carbon cycle needs to run harder lah. Okay, so what about this one? This thing that is produced. So what happens to this? So this one here is basically gonna be pushed out of your chloroplast. It's gonna be pushed out and then enter peroxisome, okay, being converted into glycolate. And then, okay, in the glycolate, it is going to be converted further to this, and lastly being added with an ammonia to form glycine. Okay, to form glycine, and lastly, it is gonna part of it generating serine. But here, as you can see, you have carbon dioxide being released. Okay, so this is the part where you lose one carbon dioxide. Okay, so unlike a normal pathway where you have two of this, okay, where you have two of this being produced, all one goes to regeneration, one goes to form sugar. But this time around, because nak buat macam mana kan, sebab oxygen dah bind, you dah generate this one with two carbon. So one of the carbon can be reused and harvested back again, but eventually the process will lead to the production of carbon dioxide. Okay, so rugi satu carbon. So which is why we lose one carbon as carbon dioxide. So in the process of dealing with this, yeah, the process here will lead to the reduction or release of carbon dioxide. Okay, because oxygen consumed and carbon dioxide released, the process is called photorespiration. Okay, so this is not preferred lah in a plant, because normally you want a plant to be producing oxygen rather than producing carbon dioxide, right? So this one leads to carbon dioxide formation. So if you consume oxygen, release carbon dioxide, it is respiration. Okay, but because it happens in plant, it's called photorespiration. Okay, wasteful process, loss of carbon as carbon dioxide without producing ATP or NADH. Okay, biochemists have been trying to engineer better Rubisco. Okay, Rubisco, it's not like Rubisco wants to bind oxygen, but it just so happens that this Rubisco has the property to bind of oxygen. So nak tak nak, kalau dah bind, so it end up generating this. Okay, so this, the cell will have to deal with this. So one of the process is gonna push and end up generating serine. Okay, some part of it, you have the ammonia component, it's gonna be forced to produce other things. Okay, so rather than generating ATP, you see the process still consume ATP some more. Okay, so which is why this is not preferred. Which is why not preferred. Okay, Rubisco itself is already having high tendency to bind with carbon dioxide, but in cases where oxygen is higher, and so you cannot prevent. It's not like there's a pegawai depan Rubisco tu kata oxygen you tak boleh masuk dekat active site. No, there's no such things like that, right? So eventually you have moments where oxygen can actually bind and interfere. Okay. Which is why different plants have different mechanism. Okay, which you are aware, different plant, which they call it like C3, C4, and also chem plant. Okay, so C3 is basically, I'm sure you have somewhat come across this before. C3, C3, C4 is basically referring to the first, okay, the first product that is produced from the binding of your carbon dioxide. Okay, in C3 plant, you have the first thing that comes out is a 3 carbon. Okay, so if it's a C4 plant, the binding of carbon dioxide will lead to the formation of a 4 carbon. Okay, if you follow through, it's that Calvin cycle bundle shape. Carbohydrate. Okay, C4 compound. So, yeah, this one. This one is the first. Okay, oxaloacetate. Okay, so the main difference between C3 and C4 is they name it according to the first product that is produced from the binding of your carbon dioxide. Okay, when you have carbon dioxide coming in, it forms a C4. Okay, oxaloacetate is a compound with 4 carbon, whereas your 3PG just now is a 3 carbon, okay, C3. Okay, so the difference between this part, the next few slides, I'm going to use a video to show you a video lah. Okay, it's easier to understand the terms. Okay, so for example, in the C4 plants, in order to avoid phosphorespiration, they have this spatial separation. Okay? Spatial separation as in separation of process. Bilik yang berlainan buat benda yang berlainan. Okay, so in C4 plant, they have different cell structure. Okay, you have this mesophyll cell and also bundle shape cell. Okay, then whereas for chem plant, okay, normally the chem plant, which are normally represented by your cactus, for example, okay, so they have a slightly different mechanism whereby they they do different thing at different time. Okay, so this one is do different thing at different place. Okay, spatial separation. This one is do different thing at different time, temporal separation of carbon fixation and carbon utilization. So how is that? So there is this animation. Okay, let me show you the video. Sekejap, saya stop sharing sekejap. For your information, there's actually a lot of this animation inside YouTube that you can find. There is some, with the advancement of animation technology, right? So some of them are really, really nice lah in a way, whereby it shows you how it makes you see that how it actually looks like inside itself. Some of them can even come up with this virtual uh, tool inside a cell, for example. Okay, so now this is just one of it. So which is a quick, straight to the point comparison between C3, C4, and also chem plant. Okay, so now let's just give this. Okay, same. Let me know whether can you listen, hear the sound in a moment. Okay, let me just. Split. Let's keep your screen straight away. Okay. Any sound? Ada bunyi kah? Boleh dengar? Can
Here.
hear?
Yes,
Can
Doctor.
hear? Okay. So just a quick 3 minutes. Okay.
Photosynthesis is the process by which plants capture the energy of the sun to make carbohydrates. But there are minor variations in how plants do this, depending on the environment in which the plant lives. In this tutorial, we'll look at the differences between the three types of photosynthesis: C3, C4, and CAM. Most plants, around 85% or so, carry out C3 photosynthesis. As we will see, the name C3 has to do with the type of carbon molecule produced during the process. C3 photosynthesis occurs in the mesophyll cells of the C3 plant, which are located just under the surface of the leaves and other tissues. Let's take a quick look at how photosynthesis occurs in these cells. During C3 photosynthesis, CO2 is taken up and transported directly to the Kelvin cycle, where it's fixed into a 3-carbon molecule, thus the name C3. If the stomata are open, CO2 moves in and oxygen moves out of the cell. But under hot or dry conditions, when the stomata are closed, oxygen accumulates, which inhibits the amount of C3 that can be made. Some plants have evolved an adaptation that allows them to be successful in hot, dry conditions. These plants carry out C4 photosynthesis instead of C3 photosynthesis. During C4 photosynthesis, CO2 moves into the mesophyll cell and is immediately fixed into a 4-carbon molecule, thus the name C4 photosynthesis. But the anatomy of a C4 plant is different from that of a C3 plant. A somewhat simplified version of this process is shown here. In a C4 leaf, chloroplasts are located in mesophyll cells, but they are also located in bundle sheath cells, which surround the leaf vein. CO2 moves from the mesophyll cells into the bundle sheath cells where the Kelvin cycle occurs. This shields the plant from the oxygen buildup that would otherwise occur when the stomata are closed during dry conditions. A second way of addressing the problem of oxygen is to change the timing of photosynthesis. This is called CAM photosynthesis, and in many ways it's similar to C4 photosynthesis, except that the timing of the process changes. During CAM photosynthesis, CO2 is taken up during the night when the stomata are open and is fixed into a 4-carbon molecule. It's then stored until daylight as an intermediate molecule, which gives the CAM cycle its name. This adaptation allows photosynthesis to occur in drier climates than in both C3 and C4 photosynthesis. A great example of a CAM plant is the cactus. Let's do a quick review. During C3 photosynthesis, CO2 is taken up and transported directly to the Kelvin cycle. The C4 pathway alters the location of photosynthesis, and the CAM pathway changes the timing. Both of these adaptations allow some plants to survive in environments that are less than ideal for the typical C3 plant.
Okay. So basically, that's a straightforward.
Introducing the new Dutch.
Okay. Straightforward comparison between a C3, C4, and chem plant. Okay. So there's actually another one which you, if you are free, you can go and have a look. Like this one more. this one. We can click on this one, but the animation is not so advanced, no. Okay, as you can see here. So there are several many more, okay, which you can take a look. Okay. Some of them even, like for example this one. This one is more towards the anatomy lah. Okay, but you want to look so that we can have a clearer view what actually how does actually a plant, the inside of a plant looks like, or the leaves of a plant looks like. So feel free to, if you are free, go and have a look lah. Okay, so basically that is all for today on photosynthesis. Sure, we have covered both two parts. Okay, we have your light reaction, we have your dark reaction. Okay, so the important things that you need to know, okay, from photosynthesis is, okay, make sure you know the difference between your C3, C4, and CAM plant. Okay, what are the difference? Okay, so where does all those light reaction and dark reaction take place? Okay, so they are both at the located at different location. And what are the important things produced from each stage? Okay, what are needed and what are the things that are produced? Okay, so those are some of the few things that you need to know. Okay, for okay, for photosynthesis. Okay, jadi now, terkejut tadi ke? It's a baby's sound, right? Just kind of like something to keep us all refreshed lah. Okay. Um, okay, so far that is all for today. So any questions so far, anyone? Okay. Just now you all have taken your attendance, right? And I noticed that there's someone inside the chat requesting for, hey, 335. Oh, so this one is expensive, okay. Yes, okay. Okay, oh, so this one, okay, so let's see the slide. Okay, okay, okay. Terkejut saya ingat for the past 1 hour you all cannot see my slide. Okay. So any questions apart from attendance? Attendance, I noticed that there's one person requesting for Muhammad Azri. Okay, you requested for another attendance from group 3, no problem. I'll help you for that. Okay, so if anyone needs a manual attendance, feel free to type your name inside the chat box. Okay, so any questions? If no questions, basically that is all for today. I will see you all again next